Hi guys, hope you all are well. Welcome to my channel Sutapa's Biology and I am Sutapa Datta. My today's topic is biomolecules. Lastly, I have completed enzyme and this is the first video of biomolecule. Enzyme is also considered in biomolecule, but it's the very specific one and unique one. That's why I have given the different video for this. And now about the other biomolecules. Now what is biomolecule? How can we decide that it's the biomolecule? Actually, the carbon oriented all the compounds which are present in the living tissues are known as biomolecules. So what is biomolecule? All the carbon compounds which are present in living tissues are known as biomolecules. Now, biomolecules are two kinds or two types. Number one is micromolecule and number two is macromolecule. Now in micromolecule, we can consider those biomolecules which have less than 1000 molecular weight. Okay, less than 1000 molecular weight, all the compounds are known as micromolecule. Less than 1000 molecular weight, and macromolecules are more than 1000 molecular weight. In some books they have used the unit Dalton, but now it's not considered. The Dalton is not the unit of the molecular weight, as molecular weight is the comparative amount, so it has no unit. Now in micromolecule and macromolecule, we can consider all the organic compounds like that carbohydrate, protein and fat. These are the primary metabolite. They have another name, that is they are considered as primary metabolite. Other than this, there is some other metabolites which are obtained from the different plants, fungi, microbes, etc. Those are known as secondary metabolite. They have different kinds, at least 1000 types of kinds are there, and they are known as the secondary metabolites. So what are the secondary metabolites? Other carbon compounds are obtained from plants, fungi and microbes. They are secondary metabolite. Now secondary metabolite have different kind like that. Secondary metabolite. The secondary metabolite have antibiotics, alkaloids, antibiotics, different kinds of that. Alkaloids like reserpine, like thein, caffeine, etc. Then gum, resin, etc. These have many economic importance. All these secondary metabolites, they have many economic importance, but in our syllabus or in your syllabus, it's not included. So I'm not giving more details about this. In your higher classes, if you study with this biological science, then you will get it. So this is about the primary metabolite and the secondary metabolite. Now we shall discuss about the different types of primary metabolite. As already I have told you that secondary metabolite is not in your syllabus. Okay, so this concept is required for your competitive exam, so I have given that. So in case of the primary metabolite, the main components are the carbohydrate, protein and fat. Now in case of the protein, the amino acid is the unit. The unit of the protein is amino acid, and in amino acid there are different kinds. We know that the 20 types of amino acids are present in our body. In human body, 20 types of amino acids are found. So we are starting the primary metabolite. And amino acid is included in that, that is protein. Protein has the unit amino acid. Now in amino acid, the main components are the amino group and carboxylic group. The bond formed in between the amino group and the carboxylic group is known as peptide bond, which form the polypeptide chain. By formation of the peptide bond, the amino acid gets associated with each other to form the polypeptide chain, and that polypeptide chain is ultimately that forms the protein. Okay, so the basic thing of the protein is amino acid. Now, the amino group and carboxylic group of the amino acid are ionizing. Ionizing highly. So that's why in case of the different pH, when pH is different, then the structure of the amino acid get changed due to this ionizing characteristics. Now in amino acids, we can divide them into three types. What are the types? That is acidic amino acid, basic amino acid and neutral amino acid. Now what is acidic one? In any amino acid, the functional groups are of two kinds. That is, already I have told you, that is the amino group and carboxylic group. Now, if in any amino acid, the amino group is more and carboxylic group is less in comparison to that, then the amino acid will be basic type. It's the basic amino acid, basic type. Example, valine. And another one is where amino group is less than the carboxylic group. So carboxylic group is more, then it's known as the acidic amino acid, acidic type. Example is glutamic acid. And number 3 is where amino group and the carboxylic group both are equal, then it's known as neutral. And example is lysine. So these are the different types of amino acids, and this is about the protein. Now protein has different structure like that: primary structure, secondary structure and tertiary structure, ultimately the quaternary structure. It has 4 types: primary, secondary, tertiary and quaternary structure of the protein. Next is about the lipid. The unit of lipid is known as triglyceride, TG. The unit of lipid is known as TG. TG or triglyceride. And it has, the triglyceride has ester bond. They have ester bond between them, and in a triglyceride, if we analyze it further, then we shall get one glycerol is attached with 3 fatty acids. Plus 3 fatty acids. Lipid is very important for the living organisms, and all the living cells, they have the bilayer of the lipid in their cell membrane. So lipid is very important, and that lipid is known as phospholipid, where one of the fatty acid is substituted by the phosphate group. That is the phospholipid. So in cell membrane we get phospholipid. Very important information for you. So if we break the lipid, then we shall get glycerol and fatty acids. And on the basis of the structure of the lipid, there are 2 kinds, that is the saturated and unsaturated. On the on that basis, they have oil and butter, etc. Butter, ghee, etc. Now, in case of how it's related with the formation of oil and butter, ghee. If in structural formula we can get that the more they have unsaturated lipid, that is oil. Their physical the property like that, the oil remain liquid at the room temperature. And what about the saturated lipid? That is the butter. In saturated lipid, that is butter or ghee, etc., we know they remain solid or semi-solid at the room temperature. Now, this is the difference in their physical characteristics. But in chemical characteristics, what is the unsaturated lipid? If they have one or more than one double or triple bond. So they are unsaturated lipid, and in case of the saturated lipid, they have only single bond. So by hydrogenation, that is the character of the lipid, by hydrogenation we can convert the unsaturated to saturated one. And that's why the vegetable oil can be converted to dalda, etc. Vanashpati. This is known as hydrogenation. Conversion of unsaturated to saturated is known as hydrogenation. Then about the carbohydrate. Next we are coming to the carbohydrate. In carbohydrate we shall get the glycosidic linkage in between the units of them. What is the unit of the carbohydrate? That's the monosaccharide. Example is glucose. In carbohydrate, basic unit is monosaccharide. The monosaccharides remain attached with one another to form the polysaccharide, and in monosaccharide there is single unit like glucose. If there are two units, that is disaccharide. Example of that, maltose, where two glucose molecules are attached. Glucose plus glucose. And in case of the oligosaccharide, they have more than 3 and less than 10 units. They are known as oligosaccharides. In our cell membrane or in a living system cell membrane, they have the oligosaccharide in the cell membrane. And they have glycosidic linkage. So this is about the preliminary or very basic discussion about the primary metabolites, and that's all in this video, and in our next video we shall continue the topic.